So to pick up where we left off, you guys discussed last class transcriptional regulation to kind of complete the entire process of transcription, the entire series on transcription, I should say. And today we're going to try and complete the last component of central dogma and talk about translation. And we're going to be talking about it within the context of a molecule called ricin that some of you may or may not have heard or be familiar with. Ricin originates from the castor plant. It's this colorful plant here that you can see depicted on the slide that possesses these castor beans. What's important to note is that there's an extract that you can collect from the beans called castor oil. That's something you may have all heard before. It's been used for many different purposes. It can sometimes be used to help relieve some digestive issues. It's been used for as an ingredient in some plastics. But what's important is that when you take the extract and separate out the castor oil, the insoluble part, there's a soluble portion that's actually quite toxic. Again, it's not the castor oil that's toxic, but rather it's when you remove the oil from the extract and are left with this like water soluble portion, that's the actual toxin that we've called ricin. You may have been familiar with it. It's been used as it's been depicted in a lot of movies and TV as a toxin of choice. But it's actually quite lethal in fairly low quantities. It only constitutes a small fraction of the actual castor bean itself. But what ends up happening is exposure to small amounts of ricin can actually lead to pretty severe vomiting, really severe diarrhea, severe dehydration, can actually lead to a decrease in blood pressure to an extent that's so degree that, again, this touches back on something else we've talked before, can actually lead to shock, where you can't perfuse your organs sufficiently, and death can occur on the order of a few days. There have been some really high-profile assassinations where ricin has been used. You've probably seen it depicted in a lot of media where there's an umbrella with a toxin on the tip, where someone is pricked with it and then they eventually die. That's actually based off of a high-profile assassination of a USSR dissident who was assassinated, and only the toxin that he was exposed to was only on the order of about 200 micrograms. So it's a really potent toxin, and the reason why we are discussing it is because its mechanism of toxicity relates to translation. The way ricin works is that it'll destroy cells by permanently shutting down the ribosomes, a key component of translation. And so we're going to circle back at the end after we talk about translation to try and understand and describe how ricin works and how it actually tries to shut down our ribosomes. So what we're first going to try and do is define some key codes here that'll actually bridge together some things that we've talked about over the course of this unit, as well as some things that we talked about in the very first unit from the very first lecture. We have in our mRNA, we have our specific ribonucleotides, we have our A's, our U's, and C's and G's. And the genetic code whereby translation reads our mRNA is that it's reading three ribonucleotides at a time. And we call these three ribonucleotides at a time codons. It's this triplet code that is how three ribonucleotides will actually encode for a specific amino acid. Because we have 4 possible ribonucleotides, and because a codon, again, this like triplet code that encodes for an amino acid, because there's 3 of them that are within this reading frame, for lack of a better phrase, we have 4 to the third or 64 possible combinations. Now, if you've all can think back to our amino acids lecture, bless you, we don't have 64 possible amino acids. And so what's important to note is that we are going to talk about why that's important, why there's not, uh, despite having 64 possible combinations, why it seems to be that there's a degree of redundancy here. But what's important to also note is that we have 3 codons here, again, these, or 3 triplet code, where they actually will encode for a stop codon, meaning it'll be what encodes for where translation will stop. But what's also really important to note is that there's a specific codon. Sorry, I think I'm going to go back. I'll jump to the next slide there. There's another specific codon that I do want you guys to memorize. I'm not going to ask you guys to memorize this entire table. Again, I'll leave that to the biochemistry class you guys will take in the future. So a key, the redundancy that I talked about, where there's 64 possible combinations, but it only encodes for, but we only have 23 possible amino acids. Why that redundancy in the system is important. You can look at the table and see that there's some redundancy where different codons can encode for the same amino acid. That redundancy is important. But key features of this genetic code that I want you guys to take away and focus on is that there's a degenerate quality to it where an amino acid can be encoded for by more than one codon. So if we look at leucine, there are lots of combinations of codon, or lots of codons that can encode for leucine. That's the degenerate quality of our genetic code. Another important quality I want you guys to take note of is that it's unambiguous, where despite there being a redundancy, each specific codon will always encode for a specific amino acid. So for example, the codon CUU will always encode for leucine. It won't encode for some other amino acid. So this is the unambiguous quality of our genetic code. It's also non-overlapping. What that means here is that during translation, there's a specific reading frame of 3 bases at a time. So that's why we've dubbed specifically 3 bases at a time as codons, because that is the reading frame within which translation is occurring. And again, we have 64 possible combinations, but only 61 of them encode for an actual amino acid. The other 3 encode for what we call a stop codon. A stop codon is a key signal that stops the growth of a polypeptide during translation. Again, I'm not going to ask you to memorize all of these, but one that I do want to try and draw your attention to is what's called the start codon, AUG. It encodes for methionine. And so that codon is important for you all to take note and know. And one helpful trick to help remember that one is, since it's AUG, you can think of it, since it's AUG and it's a start codon, you can think of it as school starts in August. And so that is the one codon I do want you guys to memorize. We're going to take a moment to try and go over the big picture of translation, and then we're going to take time to try and dive in and fill in the details. And so, especially given that, I'll take a moment to hit pause here. A lot of this unit has been focused on processes. It's easy to get lost in the details and in the nitty-gritty of these processes. What I want you to try and do is to try and think about what's the big picture, what's the whole story. And then once you can grasp the whole picture, then you can try and fill in the finer details of perhaps what molecule or protein is serving this role during this process. What helps to stop this process? What helps to kickstart it? And so again, try and grasp the big picture. That's what these first, for each lecture during this unit, we're trying to have a slide that encapsulates the big picture, and then we jump into the finer details and tie it all together. So I just want to help provide that direction, especially as we have the exam coming up on Thursday. So for the stages of translation, we talked about the different species of amino acids. All 3 species of, sorry, I said amino acids, of RNA, forgive me. We talked about the 3 different species of RNA. All 3 species are going to be present and acting during translation. So at the beginning of translation, we're going to have a transfer RNA that's going to be carrying an amino acid. And we're going to have these ribosomal subunits that come to get together to form a ribosome. They're going to be during the process of initiation, they're going to form this complex at the start codon, at the first codon that'll be read. And we have the amino acid here, methionine, because that methionine goes to the start codon AUG. From there, we're going to have subsequent amino acids brought in by transfer RNAs that add those amino acids onto this growing peptide. And finally, at termination, what we're going to have is at our stop codon, they are not going to prompt they're not going to bind to a transfer RNA that's carrying an amino acid, but rather at a stop codon, we're going to have a protein that's called a release factor that's actually going to be carrying a water molecule that's going to help with the formation of a carboxyl group to create the C terminus of that peptide and promote the cleavage and release of that peptide to complete translation. So we talked about transfer RNAs really briefly before. We're going to take a moment to try and dive a little bit deeper to try and understand their structure and what their role is. So we've also talked about how with the formation of mRNAs that there's a hairpin loop that's a critical signal for the termination of transcription. And we briefly discussed how different RNA molecules can have different large structures. And so here with transfer RNA, we have multiple loops here that are actually pretty critical. But there's two structures that I want to draw your attention to here. At the top here, we have what's called the acceptor stem at the 3' end, where that'll actually be the site at which amino acids will bind to the transfer RNA, and there'll be an ester bond between the two of them. That ester bond is going to be key because that's actually going to provide some of the energetics for a key part in translation down the road. So we'll revisit that ester bond later on, but just to take note that the energetics with that ester bond are going to be key later down the road. Next, I want to draw your attention to this loop here where we have what's called the anticodon. The anticodon is what's going to be complementary and anti-parallel to the codon during translation that it corresponds to. And so, again, when we're looking at our transfer RNA, and when we're trying to determine which ones correspond to the codons, it's this exact 3 base sequence here that's dubbed the anticodon that's going to determine which transfer RNA is binding to our codons on our mRNA molecule. And when we look at this in actual 3D tertiary structure here, these are what these loops look like. But again, we have our anticodon and our acceptor stem where our amino acid is attaching to our transfer RNA. And the mRNA is underneath there. There's a key feature. Yes, go ahead. The acceptor stem is at the 3' end of the transfer RNA, so let me take a step back. The main role of transfer RNA is to deliver amino acids during translation. The 3' end is the site at which transfer RNA is bound to that amino acid. And so we just call that site the acceptor stem because it's accepting the amino acid there. Within the anticodon, there's actually a really unique quality of that anticodon, specifically one of those bases. So as I mentioned before, we have one transfer RNA for every amino acid. And we don't have 61 possible amino acids as we have 61 possible codons. And we don't have 61 possible transfer RNAs. So what's important to know is that there's actually some flexibility within one of those bases on the anticodon. It's the first base within the anticodon. We call that the wobble position. The wobble position is the first position where the binding of complementary base pairs is less strict. There's a little flexibility with how it binds. And so because of that, multiple codons can be recognized by one transfer RNA. So when we look at and again, what's key to note is the anticodon is anti-parallel to the codon. So the first position on the anticodon is binding to the third position on the codon. Again, it's anti-parallel. So for example, the way in which what this flexibility actually looks like is we still have A's binding to U's and C's binding to G's, but once we have when we have uracil or U's on the anticodon in that wobble position, the first position on the anticodon, it can actually bind to two possible bases. It can bind to A's or G's, and once we have guanines, it can bind to C's or U's. There is a ribonucleotide that's unique to transfer RNAs called inosine that can actually bind to three possible bases there. And so the key thing to note is that the redundancy that we briefly talked about on the genetic code slide is enabled by this wobble, by this flexibility, by this not strict binding that again creates and enables this redundancy in the system. So to take a quick to have a quick slide about inosine, it's only found in transfer RNAs, and it's typically only in the first position on the anticodon. Because we typically don't have flexibility in those other bases within the codon, positions 2 and 3, it's typically only found in the wobble position. And so as an example of this, within the anticodon, we have in the first position, we have inosine and we have A and U. The codons that would be complementary and anti-parallel to this would be three possible codons. And so this creates redundancy where all of their differences are at the third position. It's AUU, AUC, and AUA. And so this flexibility creates redundancy that is going to be actually key for mutations that we'll talk about later in the slide. But it's, again, the key things to note here is that the flexibility in the wobble position and the ability of inosine to bind three possible base pairs creates flexibility that enables the redundancy that we see in the genetic code. I'm going to stop and give space for a couple questions here. Mm-hmm. Can you say that one more time? Uh-huh. Great question. So the question is about the binding of the anticodon, whether it binds to the start codon first or to the large subunit. We're going to have an entire set of two slides that's going to go into deep detail about that, but hold that question. Yes. The question is about if the wobble is only restricted, the wobble and flexibility if it's only restricted to inosine. It's not. So uracil and guanine here also have flexibility. Inosine has the greatest degree of flexibility in what it can bind to, but at the wobble position, the first base in the anticodon on the transfer RNA, that flexibility is also conferred to two other base pairs, uracil and guanine. Yes. The question is if inosine is typically present outside of the wobble position, outside of the first position. I have not seen an instance where that's the case. To my knowledge, like the current dogma in biology is that it's restricted to the first position, to the wobble position in the anticodon. All right, so we're going to go ahead and press on. So as I described really briefly earlier, the role of transfer RNA is to deliver amino acids during the process of translation. One way in which, and we talked about, each transfer RNA is going to be specific to a specific amino acid. And so one of the ways in which that happens is there's a class of enzymes called amino acyl tRNA transferases. This is a big name, but again, with enzymes, if you just look at and break down the name of it, you'll actually see what its actual role is. Amino acids, tRNA transferases. It's transferring amino acids onto transfer RNAs. And so we have one specific transferase for each amino acid because there's an actual binding pocket for amino acids, and it's going to be specific to each amino acid, to each species of each unique amino acid. And what we're going to have is that there's going to be the transfer RNA that's going to be loaded in, and there's going to be the hydrolysis of one ATP to complete the ester bond between the transfer RNA and the amino acid. And again, the energy of this ester bond is going to be really key later on when we're creating peptide bonds between adjacent amino acids on the growing peptide during translation. But a key thing to note, and it's in blue here, is that we're going to have one specific transferase for each amino acid. So when we consider what the mRNA looks like from the perspective of a ribosome, and again, going back to the overview slide, we have the ribosome that's going to be creating this reading frame where it's going to be reading each codon and shifting down one codon at a time. When if we consider what the actual mRNA looks like at the molecular level, we've talked about how we have between the we have the specific coding region, but there are regions outside of that that are actually pretty important for translation. Upstream from the start codon where translation occurs, we have the 5' untranslated region. It's typically referred to as the 5' UTR. So that's everything to the left here of the start codon. What's really key to the 5' UTR is that it's really important for the kinetics of translation and the regulation of translation. And we'll see what that looks like when we dive deeper into initiation of translation later on. When we look at the 3' untranslated region, that's everything to the right of the stop codon. The 3' untranslated region, or the 3' UTR, typically what's important for that is that it's critical for post-transcriptional stability. And so we talked earlier about how that region is really important for providing the cues to promote stability for our mRNA molecules here. And everything in between the start codon and the stop codon is our coding sequence. It's everything that encodes for the actual protein that's going to be synthesized. Typically in the eukaryotic mRNA, that's going to encode for one specific gene and one specific polypeptide. Last class, you guys talked about polycistronic genes and how there's going to be multiple genes encoded within this coding sequence here. And so one other key player that I want to take a moment to describe here is the actual ribosome itself. Each ribosome is comprised of two subunits. We have the large and the small subunit. And in addition to that, it's also comprised of ribosomal proteins that are pictured here in purple. Forgive me if I got the color wrong. Again, I'm colorblind, and I'm just going to say what I see. Forgive me if it's wrong. We have the ribosomal proteins here in purple, and then the ribosomal RNA that's going to be depicted in a lighter shade of purple. I'll let you guys correct me later on. But the key thing to note here is that there's 2 degrees of distinction here, where we have the large and the small subunits, and the ribosomal RNA and the ribosomal proteins. And so this is where, when we talk about one species of RNA, rRNA or ribosomal RNA, this is where it's present. It helps create the formation of the ribosome. Yes? The question is, does the coding sequence include the stop and start codons? So that's a great question. So the start codon will encode for a specific amino acid, methionine. That's always going to be the first amino acid. So it encodes for that. But and since we talked about this really briefly at the beginning, the stop codon will typically encode for a release factor that is not carrying an amino acid. So there's not an amino acid that's encoded for by the stop codon. So I'm going to take a second to describe bacterial ribosomes, prokaryotic ribosomes, and then I'll describe eukaryotic. So the bacterial ribosomes are broken up by their large and small subunits are typically described as being the 50S and 30S subunits. This is not having to do with their molecular weight, but it's more so rather to do with how they precipitate out when you spin them down at really high speeds. So for example, it's the large subunit that's 50S, the small subunits that's 30S. The entire ribosome complex is described as being 70S. So it's not like a clean additive quality of the ribosomes. The S stands for Svedberg units. That's the actual unit where they're determining how they precipitate out when you spin them all down. For eukaryotic, we have the large subunit is 60S and the small subunit is 40S. Together, it's 80S. So again, it's not clean molecular weight distinctions, but just the way in which they were defined historically. For each of our ribosomes, there are 4 key sites that I want you guys to take note of. One is the mRNA binding site. That's right where my mouse. That's where the mRNA is able to slide in and be available to the ribosome complex during the process of translation. When we look at the ribosome complex, when the small subunit, large subunit are together, there's 3 specific domains that you can kind of see in the top image here. They're called the A site, the P site, and the E site. Okay, so the A site, the far right compartment, is where a new transfer RNA that is loaded or considered active with an amino acid on it. The P site is the middle compartment where the polypeptide or the growing polypeptide is going to be present. The final site is the E site. You can call it the exit site or empty site. That's the site where you're going to have a transfer RNA that's going to have that's not going to have an amino acid on it, and that's going to be where the transfer RNA is going to be leaving the ribosome. So again, to try and help you try and remember what the A site and P site and E site are for, it's kind of in the not acronym, but in the nomenclature itself. A is for, you can think of it as A is for activated or amino acid. That's where the loaded transfer RNA is going to be bringing in the amino acid. The P site is where the polypeptide is, where it's going to be adding on to the peptide, and E site is the exit or empty site there. So we're going to take an opportunity now to try and dive into each part of translation to try and understand who the players are and who's contributed and what each part of translation is facilitated by. So there's going to be a key class of proteins called initiation factors. They're going to be really helpful in where they dock onto the small subunit at these sites called ribosome bindings, where they're going to be enabling the small ribosome to bind to these ribosome binding sites. In the prokaryotic system, they're called Shine-Delgarno sequences. They're in the 5' UTR. Again, 5' UTR is everything upstream from the start codon to the left of it. And once the initiation factors are bound to the small subunit, they're going to help also enable the first transfer RNA to come in and bind to the start codon. Once that first transfer RNA that's carrying in prokaryotic formulated methionine, and once we have the initiation factors there, and these initiation factors are going to have a GTP molecule on there that's important. You can kind of think of this as like an entire pre-initiation complex. That GTP is going to be really important because it's going to help with the completion of the ribosome assembly and bringing in, helping the large subunit to bind to the small subunit, forming the full ribosome complex. And so all of this is going to all of this is what constitutes initiation. And that formulated methionine transfer RNA is going to be loaded in the P site here because it has the first amino acid. this is what it looks like within the prokaryotic system. In the eukaryotic system, there's a couple key distinctions. Number one, our first amino acid is not a formulated methionine. We have a methionine. We have different initiation factors by name. They're EIFs, eukaryotic initiation factors. We don't have these ribosome binding sites. We don't have these Shine-Delgarno sequences. Those are unique to the prokaryotic system. Instead, what happens is we have the eukaryotic initiation factors that'll bind to the small subunit, small ribosome subunit. And there's going to be what's called a forward search where they bind at the 5' cap, and they're going to do a forward search to try and find the start codon. So we'll start all the way at the far end of the 5' end and move forward until they find the start codon. And this is going to be with the elongation the eukaryotic initiation factors. This is going to be with the transfer RNA methionine and the small subunit. And once we get to the start codon, that's when the hydrolysis of that GTP is going to enable the binding of the large subunit to the small. And that's when we have the entire ribosome complex with that first transfer RNA, that's methionine, at the start codon now. So the way I would describe this perhaps is that the three key distinctions is that we don't have formulated methionine, we have differently named initiation factors, and we don't have these ribosome binding sites, but instead this forward search from the 5' cap down to the start codon. So once we have translation, oh, sorry, yes, go ahead. Yeah, so because we don't have these ribosome binding sites within the 5' UTR in eukaryotes, instead, this pre-initiation complex of the eukaryotic initiation factors, of the transfer RNA loaded with methionine, and the small ribosomal subunit, all that entire complex together will start at the 5' cap and move forward until the start codon is found. Once it is found, the initiation factors, what's common between both prokaryotic and eukaryotic is that the initiation factors have a GTP. So that GTP, its main role is to enable the binding of the large subunit to the small subunit and the formation of that the full ribosome. Does that help clarify? Yes, great question. So yeah, the forward search is is by one codon at a time, by 3 bases at a time. And again, if I didn't say this, forgive me, but when we at the bottom of this image here, if you look at the size of the A site, the P site, and the E site, their size is one codon. Their size, their reading frame, for lack of a better phrase, is 3 bases along the mRNA molecule. So that's by nature of the size of ribosomes, the sites of the sizes of the sites in a ribosome. That's why we've defined codons as this triplet code, because that's how ribosomes are reading our mRNA. Thank you for that question. I appreciate that. So to take a moment to describe elongation, this could be after the methionine is there right after initiation, or this could be a much longer peptide present in the P site. But we have, as we had with initiation, where we had initiation factors, here we have a class of proteins called elongation factors, and there's 2 of them. And we're going to talk about what their roles are here. So to start off an elongation, at the very top, I would say at 12 o'clock here, we have the full ribosome with a transfer RNA in the P site that would either be the first methionine or a much longer polypeptide. What will end up happening is that elongation factors bring in and deliver a specific class of elongation factor called EFTU. It brings in the transfer RNA that's loaded with an amino acid to the A site. And the way you can remember that it's EFTU that brings the transfer RNA here, you could think that it brings it to you. This elongation factor is bringing it and delivering it to the A site. And there are 2 GTP molecules present on EFTU. The main role of the energetics when those GTP molecules are hydrolyzed is that they are enabling the binding of that transfer RNA into the A site that corresponds to that codon that's visible within the A site. Once that GTP is hydrolyzed, EFTU will leave. And so now at this point, we have a transfer RNA that is loaded or activated with amino acid in the A site. We have a transfer RNA in the P site with our either first amino acid methionine or a much longer peptide, and an empty E site. There's going to be another elongation factor called EFG. It also is going to bring in a GTP molecule. And it's important for us to take inventory of the energetics here. So EFTU is bringing in 2 GTPs, EFG is bringing in 1. EFG's main role, this elongation factor, what its main role is here, is that it's going to provide the energetics to shift the entire ribosome down 1 codon. So another way to help remember what these elongation factors are doing is that you can think of EFG as the G stands for gear, because you can think of it as a gear that turns and shifts the entire ribosomal protein complex down 1 codon. And when that happens, what was in the A site is now in the P site, and what was in the P site is now in the E site. One thing that I did not describe that I should have described is that about at 6 o'clock here in this image, before there's this shift of the whole entire ribosomal complex, that transfer RNA that came in and was delivered to the A site, it has that 1 amino acid. The energetics from the ester bond between the transfer RNA and that amino acid in the A site, that ester bond is going to provide the energetics to create a peptide bond between the peptide in the P site and this amino acid that was delivered in the A site. So what's going to end up happening is that when that peptide bond is formed, it's going to transfer the amino acid into the A site. So now when elongation factor G is functioning as a gear and shifting the entire ribosome down 1 complex, the peptide that was delivered into the A site, it's now present within the P site. And now this empty transfer RNA is going to be in the E site. So once we have the empty transfer RNA in the E site, it's going to be discharged and it's going to be recycled and reloaded by the specific transferase that corresponds to it. And we're going to be back at 12 o'clock here, where we have a growing polypeptide in the P site and the empty A and E sites. So that was the entire process. I just want to try and repeat it and zoom in a little bit and take it a little slower, just because I think that was a lot of alphanumeric soup. So we're starting here at 12 o'clock. We have either the first amino acid or a chain of amino acids, a polypeptide chain, that's in the P site. EFTU is the elongation factor that is delivering a transfer RNA loaded with amino acid. Again, EFTU, the way to remember it is bringing the transfer RNA to you. So the energetics of this GTP is going to be once that those 2 GTPs are hydrolyzed, it's going to deliver that transfer RNA into the A site there. What this looks like depicted is that we have at least in the prokaryotic system, we have our 50S and 30S subunits with our A, P, and E sites depicted here with transfer RNA. That GTP, those 2 GTPs are hydrolyzed. So now EFTU will leave, and this transfer RNA is successfully delivered because it's complementary and anti-parallel to this codon. The energetics of the ester bond is going to facilitate the formation of a peptide bond between these 2. And so now once that happens, the amino acid chain in the P site will be passed over to this amino acid, and then it's going to what's going to end up happening is that EFG, G stands for gear, is going to use 1 GTP molecule to try and shift all of this down 1 codon. Oh, forgive me. Shift all this down 1 codon, so we're back at the top of the cycle. So what this looks like in an illustrated format is we have a ribosome with a single amino acid in the P site, or sorry, Yes, one single amino acid in the P site. We have EFTU that's going to be delivering transfer RNA that's going to be complementary and anti-parallel to that codon. There's going to be a sampling here, but once the right transfer RNA has been brought in, the two GTP molecules will be hydrolyzed and the EFTU will leave. The amino acid in the P site will be passed over and a peptide bond will be formed between these two. And again, the energetics for the peptide bond comes from the ester bond between the amino acid and transfer RNA. EFG will come in with a single GTP molecule that'll be hydrolyzed and provides the energetics to shift this entire process down one codon. And once we have an empty transfer RNA in the E site, E for empty, it's also E for exit, because now that transfer RNA will exit and we're back at the top of the cycle here. So we have, at least for your studying purposes, we have all of this written down as well. But because of how much that was, I'm going to stop here and give space for a few questions. Yes. Yeah, I'll do each. Go ahead, do this. Yeah, I'll go down, but I'll walk through the GTPs and then the ester bond energetics as well. So the first GTP, the first two GTPs are brought in by EFTU. When those are hydrolyzed, it's going to be, those energetics will be used to insert and deliver the amino acid transfer RNA complex into the A site. And that transfer RNA is going to be anti-parallel and complementary to that codon that's in the A site. The next GTP molecule that's important to note is the one that's delivered by EFG. So one key thing that we can take a step back and take note of is that each elongation factor is providing GTP molecules. EFTU is bringing two, EFG is bringing in one. But they're both going to be delivering, they're both going to be using GTP molecules to enable their function. So EFG is going to be coming in with one GTP molecule, and that one molecule is going to be key for it serving as a gear to shift the entire ribosome complex down one codon. And finally, it might actually be helpful to maybe use the illustration for this here. So the ester bond present between each transfer RNA and each amino acid, there's an ester bond here. The energetics of the ester bond associated there are going to be used to form a peptide bond. That's the type of bond between adjacent amino acids, to form a peptide bond between the amino acids here. And that's going to enable it to disassociate from this transfer RNA and be passed over and bind to this amino acid. So that's how we're going to have, you can think of a polypeptide as like beads on a string. So the energetics required to connect each adjacent bead on this rowing string is coming from what was present between the transfer RNA class, that ester bond. The ester bond, let's go ahead and say this again. The ester bond here between a transfer RNA and, let me take one step back. The ester bond between a transfer RNA and an amino acid, the energetics there is required to form the connections on the beads on a string here, the peptide bond between each adjacent amino acid during translation. Does that help with the inventory of energetics? On EFG? You use both GTPs are used to deliver the transfer RNA. Yeah. Yes. What's the process behind the tRNA leaving the E site? That's the first time I've actually been asked that question. At a molecular level, I actually don't think I can describe it. It's not coming to my mind. Uh, the only thing I can think of is I'll say what I suspect, and I'll leave it there. Uh, because of how complementary and anti-parallel base pairs have these bonds between them, I suspect that there's a class of proteins that helps enable the detachment of that transfer RNA in the E site to detach and leave. But I actually need to look. I'll take a further look into that, and I'll post that on Canvas in an announcement. I appreciate that question. Yeah, that was a really good question. Thank you for doing that. I saw that hand up, and then I'll go here. Go ahead. Uh-huh. Yeah, so there's I may have actually skipped over this one piece. Yeah, so there's a class of enzymes that helps enable the formation of that peptide bond, and it's leveraging the ester bond. It's going to help break the ester bond and then leverage that to create the formation of the peptide bond. Thank you for asking that. It's to try and repeat it a little more clearly for the recording. There's a class of enzymes here called peptidyltransferase that mediates using the ester bond, the energetics of the ester bond to form the peptide bond between adjacent amino acids here during elongation. Oh, I'm gonna go here and I'll go there. Go ahead. Mm-hmm. Yeah, great question. I think I said this really quickly, but it's worth repeating. Um, Somewhat similar to some of the other processes we've talked about during this whole central dogma unit. There's a bit of sampling of like trying to determine whether there's matching with transfer RNAs that are brought in. If there's not matching, they're not gonna bind, and they're gonna leave that A site. Transfer RNAs will come in. If there's no matching at that A site at that codon, they're gonna leave. Once the right transfer RNA loaded with an amino acid is complementary and anti-parallel to that codon, then the GTPs are hydrolyzed, and it's EFTU will leave and hand off that transfer RNA amino acid complex. Does that help clarify? Okay, thank you for asking that. Great question. So for the sake of the recording, when the transfer RNA binds to an anti-parallel and complementary codon in the mRNA, this shifting, is it the transfer RNA that shifts or the whole ribosome assembly? Is that your question? It's the whole ribosome assembly that shifts. The transfer RNAs, they match that codon. So they're going to stay there until it's time to leave when the assembly shifts and they're sitting in the E site now. But yeah, when EFG is shifting things as a gear, it's shifting the entire ribosome assembly down the mRNA transcript. Thank you for asking that question. I think that was a really key thing to clarify on. In the interest of time, I'm going to press on. These are all really great questions. Some that have left me stumped, but I'm not going to keep that. I'm not going to hold that against you. So again, this slide is here to help. I know everyone learns a little differently. For more visual learners, there's this slide to kind of walk through the whole process. If you want it all written out, there's this slide as well. So as elongation goes, as this polypeptide chain, these beads on a string are growing, we're going to reach a point where we're going to need to terminate this process. And again, this process stops once we get to the stop codon. The stop codon does not encode for a codon that's going to match for a specific transfer RNA, but rather it's going to promote the recruitment of a protein called release factor. So as depicted in the images here, the release factor is not going to be delivering an amino acid. but rather it's going to be providing a water molecule that's going to help create the formation of a carboxyl group and create the formation of the C terminus, the end of our growing polypeptide. And so once that carboxyl terminus is created, that's going to promote the termination of the entire translational process, and it's going to promote the disassembly of this entire complex, the disassembly of the ribosome and all of these factors present once this release factor comes in and we have the formation of the C terminus here. It bears showing here that we have these ribosomal subunits. They're a key part of translation, as you can probably figure out here. It took a long time to characterize what the ribosomes looked like. This was an image that I believe won the Nobel Prize about 40 years ago because it was because the ribosomal subunits are so critical to biology, and trying to define what their structure is and what their domains are has been an important part of molecular biology. So to try and focus on a few key pieces here, as this growing polypeptide is being synthesized, again, biology is occurring in three-dimensional space. And as these amino acids are being added on, we talked in lecture 1 how amino acids and proteins have charge. There's hydrophobic interactions. So there's going to be folding that's going to be occurring as this polypeptide is being synthesized. And to try and take note of the energetics, taking inventory of the energetics of this entire process, we had our 3 GTP molecules that we discussed about during elongation. But we also had 1 ATP molecule that was critical for the loading of amino acids onto the transfer RNA mediated by those transferases, where we have one that corresponds to each transfer RNA and each amino acid. One thing to note is that it's possible, typically we're not going to have one single ribosome on an entire mRNA transcript, but in actuality, there's going to be many times where we're going to have many ribosomes proceeding along a single mRNA transcript. And so here we have an electron micrograph where we have many ribosomes connected to one single mRNA transcript, and where they have growing peptides coming off of them. And you can kind of see there's almost like a time scale where there's not very clear peptide chains here, but to the far left, there's longer chains coming off of these ribosomes that are visible. And so you're going to have what are called polyribosomes, many ribosomes present on a single mRNA transcript sometimes. And this is going to be key for maximizing the efficiency of this entire process. We discussed before about how you guys discussed before in the previous lecture about polycistronic genes, so there's going to be multiple genes encoded within a single transcriptional unit. And so because of that, there's going to be multiple ribosomal binding sites sometimes along that transcriptional unit, along that the transcript that's going to correspond to the different operons. And so that's going to enable for simultaneous translation of those different genes present on that one single transcriptional unit. And we talked about how the 5' UTR, the 5' UTR is where typically these ribosomes will bind and then participate in a forward search to find start codon. There are some times within cellular mRNAs, and very true with viral RNAs, where there's going to be sites that enable binding of ribosomes away from the cap. Those sites are going to be called internal ribosome entry sites. Again, those are going to be sites where the ribosomes are not binding near the cap or near the 5' UTR, but binding further down along the transcript, where particularly where there might be different genes encoded later down the transcript that can be translated independently from the cap. So one thing that I referred to really briefly, but we're going to take a second to describe, is that the redundancy of our codons is pretty key, sometimes for avoiding mutational errors. And so there's going to be three key errors, and this is going to be an opportunity for us to combine or to bring together DNA, RNA, and protein here and kind of link things together and relate them to previous lectures we've had in this class. So there's going to be three kinds of mutations that are possible here. The first is called missense, where a change at the DNA level. So these are going to be DNA mutations or DNA changes that can have or potentially lead to changes at the protein level. So missense is going to be a base change at the DNA level that leads to an amino acid change. So for example, we talked about with sickle cell disease that there's a mutation that changes from glutamate to valine. And so at the DNA level, we have a thymine to adenosine mutation here. That's going to lead to a change at the mRNA level. And so this codon at the mRNA level is going to encode for a different amino acid. And so that's how we get the change, a missense change of one amino acid to the other. Another type of mutation is called nonsense. That's where one change at the DNA level will actually lead to going from one amino acid to an actual stop codon now. And so this is potentially connected, most likely connected to the NOD2 mutation we talked about during our immunology lectures, where there was a truncated NOD2 receptor, where the stop codon showed up earlier and led to a shorter polypeptide chain. And so we have an adenosine to thymine mutation here at the DNA level that leads to a different mRNA codon, mRNA base pair, and that leads to going from a leucine to a stop codon here. Now, it's key to note that the mutations here are at the middle nucleotide within this codon, middle nucleotide within these base pairs here. Now, there are mutations that can happen at the DNA level that lead to no changes at the amino acid level. Those are called silent mutations. And oftentimes what ends up happening is that these silent mutations happen at the third codon, third base pair, third base within the codon. And so here we have guanine to thymine mutation at the third codon that leads to a change at the mRNA level. Now this is key. These codons both encode for proline here. And so there is no change at the amino acid level. So this is what we'd refer to as a silent mutation. Another kind of mutation that's key to note here is called a frame shift mutation. Oftentimes, this is going to be happening, this is possibly happening in response to this can happen with non-homologous end joining. That was a repair mechanism for double-stranded breaks where there's a sampling of nucleotides to help bring in and fill in the gaps and bring those strands back together. And so in this case here, a frame shift mutation occurs where we have a guanine inserted between the third and fourth nucleotide here. So we have, what ends up happening is that the insertion here is going to shift the entire reading frame. So we're reading these codons. When you take this down from the DNA to the mRNA level, we have, again, school starts in August. This AUG codon encodes for methionine. AAG encodes for lysine. UUU encodes for phenylalanine, and this encodes for aspartate. But what ends up happening is that when you have the insertion here, that's going to shift the entire reading frame codons going from AUG to AAG here. Now it's AUG, CAA, GUU, and a lot of times these frame shift mutations can actually lead to an early stop codon during translation here. And so one key thing to note is that sometimes these mutations can actually lead to a couple of different outcomes down the road. One of which could be potentially a gain of function mutation, where, or I shouldn't say mutation, a gain of function outcome as a result of this mutation. Gain of function means that there's typically either a new quality of a protein, or rather more oftentimes as the case, is an unregulated behavior of that protein. So one example of that could be, we talked about how enzymes a lot of times will have a catalytic domain where the where the enzymatic reaction is occurring, and an allosteric domain, allosteric regulation, A gain of function might actually shut off or change the allosteric domain, where there's now unregulated activity at the catalytic domain. Another outcome as a response of these mutations could be loss of function. As the name implies, it's the loss of whatever the quality is of that protein that the gene encodes for. So to bring this all the way back to ricin, I think I'll be able to have us stop right on time. I'll take a moment to stop there for a couple of questions before we tackle ricin. Are there any questions about any of the mutations we discussed, or perhaps anything previous to that? Yeah. Yes. Yeah, so polycistronic mRNA, as you all discussed last class, that is when there's an mRNA that encodes for multiple genes along that transcript. And so oftentimes polycistronic mRNA is going to have multiple sites at which ribosomes can bind. Instead of binding, like our genes, our DNA is monocistronic. We typically have, for one transcriptional unit, for one transcript, one gene that's encoded for it. In prokaryotes, it's typically polycistronic. There are multiple genes encoded within one transcriptional unit or within one transcript. So typically, as we described before, along our mRNA, there's going to be binding of our initiation complex at the 5' cap that does the forward search to find the start codon. For polycistronic mRNA, because there are multiple genes along this polycistronic mRNA, there are multiple sites at which ribosomes can bind across that mRNA. So that's restricted to prokaryotes, not to eukaryotic mRNA. Some of those sites at which ribosomes can bind, sometimes within cellular mRNA, a lot of times with viral. The sites at which ribosomes can bind away from the cap are called internal ribosome entry sites. Does that help clarify it a little further? Mm-hmm. Yeah. Sorry? There are some instances where there can be internal ribosome entry sites in some cellular mRNAs, but it's oftentimes not the case at all. I can't really think of any, but I'll add that as an addendum to my post on Canvas. I'll look into that and add that as an addendum. Any other questions at all before we try and wrap this up? I think I can do this in 5 minutes, and then I'll let you guys go out and enjoy the warm day. So bringing this all the way back to ricin. So ricin is a heterodimeric protein. Hetero means there's two. Dimeric means there's two pieces. Heterodimeric means the two pieces are different from one another. So we have the two pieces here are ricin A and ricin B. Ricin A is the toxin. It's an enzyme. It's a toxin. But the only issue is that it cannot get into cells very efficiently. Ricin B is a lectin. It's not cytotoxic, but it actually enables entering into cells pretty efficiently. It's also an agglutinin, which typically means that it's a class of molecules that promotes aggregation. And both subunits are bound together by a disulfide bond. And the fact that it's a disulfide bond binding them together is actually pretty important. That's going to be explained a little bit later on this slide here. So in the next class, next Tuesday, we're going to start the next unit by talking about protein trafficking, how cells move proteins around either to deliver them outside the cell or to traffic them in. And so here, ricin and it will actually basically ride our the system by which we secrete newly synthesized proteins. It's going to ride that process basically in reverse. So the lectin ricin B component is going to actually help it to aggregate at the surface here by binding to some of the sugars on our cell surface here, glycoproteins or glycolipids. And it's eventually going to be internalized. It's going to be endocytosed, taken in. And so once it's taken in into an endosome, they're going to be shuttled there. And some of it is going to be, some endosomes will become a lysosome and be destroyed. Some of the ricin will actually be shuttled back and returned to the cell surface. And again, some of it will be where the endosome becomes a lysosome and destroys it. Some of it will be actually sent, will actually ride backwards into the Golgi. And it's going to process through the Golgi. Again, some of it will escape and return to the cell surface. Or some of it will be, sorry, not escape. Some of it will be delivered via vesicle back to the cell surface. Some will escape the Golgi into the cytoplasm. The ricin molecules that escape and make it to the cytoplasm are the ones that are going to be they're going to mediate the toxicity here. That disulfide bond, we talked about this earlier in the course, where there's environments that'll actually promote the formation or breaking of disulfide bonds. So once it gets to the cytoplasm, that's a reducing environment that'll actually break this disulfide bond and separate ricin A and ricin B. Once that happens, ricin A can actually mediate its function as an enzyme. And what ends up happening here is that, I just answered this question for you. I did that in the interest of time. Once ricin A and B are separated because of this reducing environment in the cytoplasm, and it separates it breaks the disulfide bond, it'll actually target adenine on the rRNA in our ribosomes, and it'll actually depurinate the adenines here. And so once that happens, this actually will inhibit the ability of our large ribosomal subunits to bind to elongation factors. So it's going to halt translation and bring it to a screeching halt, and that's going to stop translation. And that's functionally going to lead to cell death. As an enzyme, if we have one molecule of ricin A present, it can depurinate up to 50,000 RNA molecules. As a result of that, it can functionally destroy 50,000 ribosomes. And eventually, as key critical proteins are not being synthesized, it'll eventually lead to the death of that cell. Beyond being used as a bioweapon, there are some instances in which that could actually be used. It could be used as a way to try and study translation in a research setting. It actually help us understand how different proteins will bind to different nucleic acids. And finally, if there's a way for us to leverage this to try and target cancer cells, that could actually be a pretty helpful chemotherapeutic approach to try and treat cancer. If translation really caught your attention, we have Dr. Bayer here at UVA who's studying translation. With that, since it's 3:15, I'm going to call it there, but I'll be sticking around to answer any questions. Thanks, you guys.